scientist and co-director of the Bollum Institute at the Oregon Health and Sciences University. She received her bachelor's degree in biochemistry from Elmira College and later pursued a PhD at the University of Cincinnati at Cincinnati Children's Hospital under the mentorship of Nancy Ratner. And her PhD was in cell biology, so we are talking from the standpoint of a biologist here. But after her PhD, she went on toward a postdoc at Stanford under the mentorship of William Talbot. And after her postdoc, she was recruited at the Washington University School of Medicine as assistant professor in the Department of Developmental Biology. And there she rise to the ranks of associate professor with tenure, but then decided to move where she's at since 2017. And now she was named in 2018 the Neuroscience Graduate Program Director. And to talk a little bit about the research background from Dr. Monk, she has been instrumental in this institution for establishing the zebrafish as a model to study glia. And her studies demonstrate that zebrafish and mammalian glia are remarkably similar. Through genetic screens, she discovered at the adhesion G protein coupled receptors, GPR126, is essential for myelization. And she began her independent career in 2011 and has since gained recognition as a leader in the field of glial cell biology and neuron-glial interactions. Her work on GPR126 and other G-coupled protein receptors addition has helped to lay the foundation for the rapidly growing AGPRC field. And with her, we have one of the graduate students at the neuroscience program that he will be joining her to talk about the wonderful opportunities students have at their institution, which is Mateo Lopez Espejo. And he is doing his PhD at the neuroscience program, and specifically trying to understand how populations of neurons in auditory cortex represent temporal features of sound. So he's into sensory systems. That's what he enjoys. But he also is member of the Alliance of Visible Diversity in Science, which is something he'll talk about later at the end when we discuss recruitment. So thank you, Dr. Monk, for being here, and Mateo. And I'll let you start your presentation.
Great. Tab. Oops. Nope. So sorry. Do I do my entire screen?
Yes, and then
it's open.
Okay, you share. You have to click.
Okay. You see my?
Yes, it's coming. Yeah. Now, yes, and do. Wonderful.
Okay, so I can no longer see folks, but you can all see my screen. And let me see if I have a laser pointer. Great. Okay, so I can't see people anymore, but everyone can see the slides?
Yes, we can.
Great. Okay, so thank you so much for the introduction and for the invitation for Mateo and I to come talk to you today. I'll start with some of the science that's going on in my lab before we transition to talking about the graduate program and other opportunities at OHSU. So I am the director of the neuroscience graduate program, and I do work in an institute that's focused on neuroscience. And I can imagine if I asked any of you to think about a cell in the nervous system, you would almost certainly name a neuron, either a type of neuron, your favorite neuron, or neurons in general. And that is absolutely fair. Neurons are the basic functional unit of the nervous system. They fire action potentials. Synapses between neurons enable communication throughout the entire brain and spinal cord. And for well over 100 years, neuroscientists and cell biologists have focused on the neuron. But I will point out, and if you take nothing else away from this seminar, I want you to remember that neurons are not alone in the nervous system. You have at least as many glial cells in your brain as you do neurons. And in some regions of your brain, you actually have more glia than you do neurons. And in this cartoon, you can see, well, the neurons are in blue, and all the other cells in yellow here are the different types of glial cells that you have in your brain and spinal cord. This is an oligodendrocyte. This is the cell that makes myelin. My lab is really interested in the cells that make myelin in both the central and peripheral nervous system. You can also see in this cartoon, this little cell here, this is called a microglia. You might have heard of them. They're the brain's resident phagocyte, the resident immune cell. They help eat dying cells or dead cells, clear infection. They do a lot of really important things in the brain and spinal cord. And then actually the cell I'm going to focus on for most of my talk is this cartoon shows you an astrocyte. And so astrocytes are actually the most abundant cell in your brain. They're all over the brain and spinal cord. And in this cartoon, you can see them closely associated with the neurons, as well as closely associated with the blood vessels. And as was pointed out in the introduction, a lot of work in my lab has focused on myelination and the development of myelin and myelin repair, particularly in diseases like MS. But since moving to the volume a few years ago, we've become increasingly interested in these other glial cells as well, in particular the astrocytes and microglia. Okay. So astrocytes do many, many things, absolutely essential, critical roles in your brain. Without astrocytes, we would not be alive. Any mutant that lacks astrocytes is lethal. So critical for brain health, proper human health, and proper brain function. But they also do lots of, and how they do that in lots of different ways. So you might have heard of the tripartite synapse. So, we think about a synapse as neuron 1 releasing neurotransmitters that are received by neuron 2 so that signals can be propagated throughout the nervous system. But actually at every synapse, there's also one or more astrocyte processes sitting there at the edge of the synaptic cleft and listening to what's going on and reacting to what's going on. So astrocytes provide metabolic support. They maintain ionic concentrations at the right levels in the synaptic cleft. And they're there constantly surveying and listening to the neurons. And through their interactions with the blood vessels, they can actually increase the blood flow to particular regions of the brain when necessary, shown here. So there's astrocyte infeet are always also at the vasculature, regulating blood flow, regulating glucose supplies to the brain. And in really interesting recent work that's starting to come from a number of labs, including this paper, which is from Mark Freeman's lab. Mark Freeman is the director of the Bollum Institute, where Matteo and I both, where the NGP is located and where my lab is. And so there's some really cool work coming out in lots of model organisms. This paper happens to be in fly, that astrocytes can directly control circuits and behavior, modulate circuits and behavior in ways that we really didn't understand until really quite recently. So they seem to be active players in circuits and behavior. So critical for many, many different types of functions that go on in the nervous system. How do they do all of these remarkably important things? One way is by their morphology. And so I think it's fair to say that everything that astrocytes do would not be possible without this really complex and beautiful morphology that they develop over time. And this has been described as a tufted morphology or a bushy morphology or a ramified morphology. But in this image, you can see a single astrocyte. This has been dye-filled. And so here's the soma, here's the cell body. And you can see filled from the soma these sort of thick processes, but also this really dense meshwork of incredibly fine processes that are contacting hundreds of thousands of synapses at the same time, as well as blood vessels, other glial cells and neurons throughout the brain and spinal cord. So one thing we don't understand as neurobiologists is we're starting to understand all of the things that astrocytes do, but we still don't understand everything. And we don't understand how they do everything that they do. So how do they obtain this really beautifully complex morphology and development? We don't understand completely. Moreover, I think the glial field in general has largely focused on neuron-glial interactions over the past couple of decades, which makes a lot of sense. We want to understand functional relationships between neurons and glia. But we have a lot of glial cells in our brain, and we're only beginning to understand how glial cells interact with other glial cells. And so that's another, I think, major open question. One of the tools that we use in my laboratory is the Zebrafish, and I'm not sure if any of you have had the chance to work with fish, but these are a really excellent model system. So this is a larval zebrafish. It's 5 days old. This is the head, the heart is here, the brain is inside here, and the spinal cord runs the length of the fish. This is its little tail. And you can maybe tell by looking at this fish that it's pretty optically transparent. It's quite see-through, so you can see completely through these fins here. And if we had the right microscopes, you could actually see deep into the nervous system and look at the cells. Oops. So there should have been a movie, sorry. Hopefully that'll come up in a later slide. But here, I can show you a flip book movie. So these are low resolution images showing you how rapidly zebrafish develop. So maybe you've worked with mouse before and you know that the mice develop inside of the mom and that takes 3 weeks and you can't see anything. And then the pups are born and it takes another 3 to 6 weeks for them to start to do anything, interesting and for their nervous systems to develop. With the zebrafish, you have a behaving animal within a couple of days. And this movie is going to show you the first 18 hours in the life of a zebrafish embryo. So on top, there was a single cell that has divided in two, and that's where the movie will start. And these two cells, this two-celled zygote, these cells are sitting on top of this yolk that the mom laid. And so you can see this rapid cell division that occurs on top of the yolk. This is sped up. Right now, this is about 1,000 cells. We call this a blastula. These cells are going to migrate over the yolk, and in a process called gastrulation, the germ layers are being specified right now. The brain is forming, the gut is forming, the kidneys are forming. Here you'll see an eye. The neurons are being born, axons are coursing down the spinal cord, and all of that happens in less than a day. So you can get your fish to lay their eggs in the morning, and when you come into work the next day, you have a fish that is pretty functional, pretty well developed, living in this little eggshell, and then it hatches out of that egg within a day or two, and it's hunting for food. It can do really pretty complex behaviors even at that early stage. Right, so you can get hundreds and hundreds of embryos from a single pair of adult zebrafish. As I just showed you in that movie, the development is external, and so you don't have to wait for the embryos to develop inside of mom or to be born. And these larvae are transparent, and so we can see into the developing nervous system and watch in real time the nervous system develop. And a big reason that I started to work with zebrafish, actually, in addition to this live imaging that we can do, is the ability to do large-scale genetic screens as well as chemical screens. Because the fish live in the water, we can add drugs to the water and see what happens to the nervous system. You can look at any biological process that you're interested in, but of course, we're interested in the nervous system. Great. And here are the movies that I thought were on the other slide. So these are two movies taken by a postdoc in the lab and a staff scientist in the lab, Xiaoxing Li and Nanda Coelho. And what these movies show are two different glial cells, the oligodendrocytes and microglia. So in this first movie, let's start here, you see the cell body of an oligodendrocyte, and it extends all these processes, kind of like an octopus. And what you can't see in the movie are axons everywhere. But what this oligodendrocyte is doing is it's feeling around its environment, deciding which axons it should myelinate and which axons it should ignore. And by the end of the movie, you can see these little tubes And that is the development, that's the very earliest development of the myelin sheath in this animal. And here on the right, you can see a microglial cell, a single microglia. This happens to be in the hindbrain of the zebrafish. And what this microglial cell is doing is it's moving around the brain, it's looking for invaders, it's looking for pathogens, it's also associating with synapses and eating synapses that are inactive and leaving synapses that are active there. So the zebrafish, I think you can tell from these movies, I mean, this is imaging that is almost impossible to do in a mouse. And when we do imaging like this in the mouse, we have to cut a window into the head, and it's really quite invasive. But this is just a normal fish sitting on the microscope. And we can see these beautiful oligodendrocytes and microglia. But at the beginning, I was telling you about astrocytes. Curiously, and so I've been working in zebrafish now for almost 15 years. And that whole time that I've been working in fish, it's been quite controversial, actually, if zebrafish have astrocytes in their nervous system. One reason that that's been controversial is because zebrafish have another cell type called radioglia. We also have radioglia. Mammals also have radioglia, but this tends to be a very transient population in early development that's largely gone in adulthood. However, zebrafish have radioglia throughout their life. And that's in part because in zebrafish, you have the birth of neurons happen in many different regions of the CNS throughout the life of the animal. And that's just not the case in mammals. And so one reason that people thought zebrafish did not have astrocytes is because of the presence of radioglia, and people thought that radioglia were the cells fulfilling the functions of astrocytes. Additionally, when people, many different labs would look for astrocytes, one marker that was used a lot is this marker called GFAP. That's a very common marker of astrocytes in mammals, but when folks looked at GFAP staining or at different transgenes in the zebrafish to look for GFAP, what they saw were cells that looked like radioglia. So you remember at the beginning that really bushy, beautiful cell. You don't see anything like that in the zebrafish spinal cord when you stain for GFAP. But this really bothered me, and this really bothered a lot of people that work on zebrafish because we know that worms have astrocyte-like cells, and we know that Drosophila, the fruit flies, have astrocytes, and of course mammals and humans and primates all have astrocytes. So it was very unsatisfying to me and to many people who work on zebrafish nervous systems that fish would not have astrocytes. And so I was very fortunate to recruit an enormously talented postdoc to the lab, Jakoon Chen. He is a joint postdoc in my lab and in Mark Freeman's lab. And so Jakoon actually works on flies and fish to study astrocytes. So when Jakoon joined our labs about 2 years ago, he decided that he was going to determine once and for all if zebrafish do have astrocytes or not. And either way, I thought they would have them, but I was also fine if it was radioglia fulfilling the functions, but I thought that we really needed to test that one way or the other. And so all of the work, almost all of the work I'm going to show you is from this extremely talented postdoc, Jakoon. So Jakoon started by looking at a database that's available at this website, brainrnaseq.org. If you don't know this website, it's really great. You can punch in your favorite gene and see in which cell types is it expressed. Neurons is kind of a catch-all term here, but you can look at astrocytes, at oligodendrocytes, at microglia, at endothelial cells. And this was done in the lab of Ben Barris by a really great postdoc named Yue Zhang, and she now has her own lab at UCLA, so this is on her lab's website. But Jakoon started with a RNA-seq data set from Yue and from Ben Barris's lab, and he wanted to see which genes are most highly enriched in human and also mouse astrocytes. And so he came up with a list of genes. I'm not going to show you all of the data, but what Jakoon did was a expression analysis in zebrafish to see which of these genes looked like they might be astrocytes in the brain and spinal cord of the developing zebrafish. And so I will cut to the chase and tell you that this gene, SLC1A3, which is also called Glast, which is a glutamate transporter, was the most promising gene from his expression analysis. And so what Jacquin did next was to make a transgenic zebrafish. And so if you're not familiar with this technique, basically what you do, so you have the start point of a gene. What you do is you take all of the DNA before that start site. That's called a promoter region. And with this promoter region in a DNA plasmid, instead of driving gene expression like it normally would in a cell, you can get it to drive different fluorescent reporters. And so that's exactly what Jacquin did. Take this Glast promoter, this DNA sequence, to drive different colors in the cells that express Glast. And TOL2 is just another piece of DNA that helps this whole big plasmid of DNA get integrated into the genome in many different places. So we have the Glast promoter driving a myristylated GFP, so that's a membrane tag GFP, as well as a histone M-cherry. So this will mark the nuclei, and this will mark the plasma membrane. And you inject, so you saw that movie of the two-celled zygote becoming a full embryo. You can inject into the yolk of the fertilized egg, let those animals grow up, and they will express these fluorescent reporters in the cells that express the Glast gene. And so that's what Jacquin did. He made this, we call it a stable transgenic line, because when you cross it generation after generation, you get this same expression pattern. So we can see membrane GFP all throughout the brain and spinal cord. This is the cytoplasm, and these are the nuclei, and this is the merged image. So this is, the whole fish, big overview. It's hard to see cellular resolution. So another really cool trick that you can do in zebrafish is, instead of making these stable lines that you can breed over and over, you can also just take this DNA construct, inject it into the embryo, and then you can look with single cell resolution. So I'll show you that in just one second. But this is zoomed in a little bit more on the spinal cord. And you can see all of the nuclei here in the middle and these membrane processes on the edges. So you can see the brain here, shown here, and the spinal cord at different angles. Right. So are these cells actually astrocytes? So to test this and to convince ourselves, we decided that we needed to work through a checklist and prove to ourselves that these really are astrocytes. Do they look like astrocytes? Do they develop the way mammalian astrocytes do? Do they express other markers that astrocytes should express? Do they tile with each other? And I'll explain what that means. Do they associate with synapses? And do they have calcium activities like we would expect an astrocyte to have? Okay, so I'll start here. And sorry, this is the sparse labeling technique that I was mentioning on the earlier slide. So basically, instead of making stable lines, we can take this same DNA construct, inject it into the very early embryo, either into the single cell or into the yolk, and then let the animal grow up, and we'll see sparsely throughout the entire body, throughout the brain and spinal cord, single cell clones that have our fluorescent reporters of interest. And so here's a single cell in the spinal cord. So instead of everything mushing together because there are too many cells, we see a single cell. And I think you can appreciate that this isn't that long radial glial-like cell. This looks a lot like that bushy ramified mouse astrocyte that we saw at the beginning of the talk. So the nucleus and this really ramified processes external to the nucleus. So I'm going to call these spinal cord astrocytes for the rest of the talk, but I will go through the checklist to prove to both of us, to all of us, that these are in fact astrocytes based on the definition that I outlined on the previous slide. So in terms of their development, are they coming from radial glial cells? When do they begin to transform? And in fact, what Jacquin did see is before 2 days of age, these cells do look quite elongated. And right around 2 days, they start to shorten this stalk, and they transform into what appear to be astrocytes. So what I'm going to show you is a time-lapse movie of a single cell, this single radial like stem cell turning into an astrocyte. And we can do this imaging in the zebrafish. We simply mount them on a microscope slide, leave them overnight, and come back in the morning to see our movies. And so this imaging started at 2 days of age. So just about a day, that first movie where you saw the fish develop, this movie starts about a day after that. And so you can see this radial stalk is shortening while the membranes are really elaborating, spreading out, staking out a bunch of territory. And I'll play that again, because this, I think, is a real strength of the zebrafish model system, to be able to see the development of a mature astrocyte from its progenitor cell, to our knowledge that had never been visualized before. So we can see the cell transform. And additionally, another great thing about the fish is actually Jakun is able, or you, if you do the experiments, you're able to image the same cell, put the fish back in water in the morning, and then at night image that same cell again and again and again and again. And so Jakun did this over the course of a week, where you can watch this cell at 2 days of age slowly becoming, day after day, a little more mature. And then at about 4 or 5 days, the cell looks about the same as it does at 9 days. Maybe the membrane gets a little bit bigger, but the bulk of the maturation is happening between really 2 and 4 days post-fertilization, 2 and 4 days of age. And that is quantified here. So we can, Jakun can quantify the territory that an individual cell takes up, and this is how many cells he examined in, I think it was about 5 or 6 fish total. And you can see that between 2 and 4 days, the cells get bigger and bigger, and this sort of levels off by 9 days, and that's the latest that he looked. And this is just a cartoon showing you where in the spinal cord the cell bodies are located and where the really elaborate membrane processes are located. Okay, we know they express Glast. That's very important. Jakun also looked at another marker. It looked actually at several markers of astrocytes that are known to be expressed in mammalian astrocytes. One of these is glutamine synthetase, or GS, which is a very strong marker of astrocytes in humans and in mouse. There is an antibody in zebrafish available, and so Jakun did an antibody stain in his transgenic fish. And you can see very robust GS expression in these same cells that express Glast. So our astrocytes do express both Glast and GS, and I'm not showing you the data, but they express a number of other astrocyte markers. And we know this by both RNA expression analyses as well as single cell RNA-seq analyses. And so they look like astrocytes, they express genes, and they have proteins that astrocytes should have. So that's fantastic. Next, we wanted to look at their tiling behaviors as well as their temporal association with synapse formation. And so a really interesting property of astrocytes in both mammals and Drosophila is this phenomenon we call tiling. So astrocyte 1 and astrocyte 2 are right next to each other, but they really respect each other's territories. And these territories are established early in development, and they do not change throughout the life of an animal under normal, healthy conditions. And so here you can see in Drosophila, the red astrocytes are completely separate from the green astrocytes. Same thing in the mammals. But via this tiling mechanism, they cover the entire brain, and they are coupled to one another by gap junctions. And so this is the way that an astrocyte at the top of your brain can communicate with an astrocyte at the bottom of your spinal cord through this tiling mechanism from this highway of astrocytes. And so we assumed that zebrafish astrocytes would also tile, but of course we had to test this. And so to get at that question, Jakun injected a construct to make some astrocytes green and some astrocytes red. And when he looked to see a red astrocyte next to a green astrocyte, indeed they tile very similarly to what's been described in other species. Moreover, in other species, as I told you about that tripartite synapse, the astrocyte processes are right there at the synapse, listening and modulating levels of neurotransmitters and ionic balance. I also showed you that the massive elaboration of the astrocyte processes is occurring between 2 and 4 days post-fertilization. And interestingly, that's also when synaptogenesis, when synapse development is happening in the zebrafish spinal cord. So this SV2, this is a marker of synapses. The GFP and the cherry are again marking our astrocytes, either the nuclei or the processes. And then in the merged image, you can see everything. And what I think you can appreciate is that as you get more and more synapses, you also get more and more elaborate astrocyte processes. So this is correlative, but it does suggest that the astrocyte processes are very closely associated, which you can see a bit better here. So GFP is the astrocyte, SV2 are the synapses, and you can see very close association with the synapses and the astrocyte processes. So we still have a lot of work to do to prove, and we can do more high-powered imaging, we can do electron microscopy, but we think this is indicative of association with synapses as we see in other species. So this is all great. And now I'm going to show you, I think, what is my favorite data that came out of these studies. So I told you that that neurons fire action potentials, but glial cells do not. You know, they're not really built for that. They don't have long axons. Their job isn't to communicate long distances. Their job is to communicate sort of in their local environments. But what many glial cells do, and what astrocytes in particular do very well, is very dynamic calcium signaling. So lots of folks have done really great work in mammals and in fly looking at astrocyte calcium signaling. And what we know is that we have these big calcium events, which you'll see a movie of in just a second. But in astrocytes, you also have these little whispers of dynamic microdomain calcium signaling. And this can be induced by norepinephrine. And so this paper in particular is from Dwight Burgles' lab at Johns Hopkins University, but many labs have shown similar findings. And so, you know, given the power and the transparency of zebrafish and being able to do this live imaging, Jakun made yet another very cool transgenic line where the same promoter is now driving GCAMP6, myristoylated GCAMP6, so membrane tagged calcium indicator. And this is an awake, it's not behaving because it's stuck under a microscope slide, but this is an awake animal. And every time you see a burst, this is calcium signaling in different astrocytes. So you'll see these big body bursts of calcium. And if you look closely, you'll also see these little whispers of microdomains. It's like a lightning storm. This is what's happening in your spinal cord and your brain, by the way. Right now, your astrocytes are always just whispering to each other and talking to each other and talking to other cells. What any of this means, we don't know, but it's certainly something that we would all like to figure out. But with this tool, then, we can see the astrocytes talking to one another throughout the entire nervous system in this awake, intact animal. So I think that's super cool, and I really like looking at these movies. Importantly, as I told you, the mammalian astrocytes are, these microdomains can be induced by norepinephrine, and we see the same thing here. So, you know, there's still a lot of really cool calcium events going on in the control-treated spinal cord, but in the norepinephrine-treated animals, you see many more of them. And we found a way to quantify this by collaborating with Kira Pazcancer. Her lab is at UCSF. And she developed this software called Aqua, astrocyte quantitative analysis. And so Jakun went down to her lab at UCSF, obviously before the pandemic, and learned how to quantify these calcium signaling events. And so what I'm showing you is sort of the bursts, how many calcium signaling events we get, how many you get in a minute, how large of an area they take up, how big they are, and how long they last. And so this is just a way to quantify what we saw in the movies, but importantly, these transients are basically indistinguishable from mammalian calcium transients. And so We are pretty convinced that we have found zebrafish astrocytes. They look like astrocytes, they express what they should, they act like astrocytes in many different ways. So now we can do fun science, right? So we have these astrocytes now, we can start to do genetic and chemical screens to figure out how they do what they do, and in fact, even a bit more basic, what they're doing. And so we decided as a proof of principle to look at this receptor, this FGF receptor, because we know in flies, when flies have a defective FGF receptor, their astrocytes have a defective astrocyte, sorry, when the fly astrocytes have a defective FGF receptor, they cannot make their elaborate processes. And so you've probably heard about CRISPR-Cas9, this Nobel Prize winning technology that you can get in and cut the DNA exactly where you want it to. And so there are lots of techniques in zebrafish. This is kind of a busy cartoon, but basically we can use the same Glast promoter to drive CRISPR-Cas9 in our astrocytes. And we targeted different guide RNAs. So this is where the Cas9 knows where to cut to the FGF receptors. So basically what this is a way to induce mutations in any gene of interest and any cell of interest. And in our case, we did this in the astrocytes. And so inject this construct into the single-celled animals and look at astrocyte morphology. So as you've seen, normally astrocytes have this really nice tufted morphology, but we can also perturb this by disrupting different FGF receptors. And so that's shown here. Zebrafish actually have 5 different FGF receptors, and we found that 3 of these receptors didn't really do anything to the astrocyte morphology. But if we perturbed FGF receptor 3 or 4, the astrocytes started to look quite funny. And so we're growing up these fish. We're going to make double mutants. But it's interesting that FGF receptor is conserved in zebrafish from flies. And this is just another way to look at the astrocyte surface area. And when we disrupt FGF receptor 3 or 4, it becomes reduced. But really, this is a proof of principle, because now we can go and hit any gene that we're interested in, knock it out in astrocytes, and see what happens. And so to summarize the data part of my talk, we have described for the first time astrocytes in zebrafish based on their morphology, on their gene expression and other proteins that they have, how they tile interact with other astrocytes, how they interact with synapses, how they talk to each other and other cells in the nervous system. And we've begun to genetically dissect what regulates their morphology. And this is great because now we can do a lot. Now we can do the cool science, right? We have an animal that has all the major classes of glia, just like humans do, the oligodendrocytes, the microglia, and the astrocytes, that we can see in the same animal, in the living, behaving, reacting animal. We can do light imaging. We can do many different screens, targeted or unbiased. And I'm really excited to see where this work is going to go, both in Jack Koon's lab when he starts his own lab one day, and also in my lab and for projects for my trainees. And so we're really interested in understanding astrocyte biology, kind of taking some hints from single-cell RNA-seq studies that have been done. I'm really fascinated by thinking about how glial cells interact with other glial cells. You may remember a professor named Anusha Mishra who came to visit you all a year or 2 ago, I think, and she works on astrocyte blood vessel coupling. So we're going to work with her on some collaborative projects there, and really getting at this big question of what astrocytes do to control neural circuits and behavior. And for this, we'll start with the simple calcium imaging, because it's so straightforward to do in the zebrafish. And so I will thank my lab, the folks who did all this work. So as I told you, almost all of the work was done by Jack Koon, really great postdoc in my lab and in Mark's lab. He's been helped with some of the transgene construction by research assistant in the lab, Cam Patton, and this is some members of my lab, as well as some collaborators and friends from other labs on top of Mount St. Helens last summer, which is an active volcano, but not that active because you can still climb to the top of it. This is in Washington State, just across the border from Portland. We have great collaborators at OHSU, as well as other parts of the country, and I'll thank my funding sources. So I will stop the presentation here. Maybe I'll stop sharing to do the questions, and then I'll open the slides back up in just a second. How do I? There we go. Great.
Wow. This is the reason why I love neuroscience. Oh my God, I learned so much. And also I just, something, we always are so neuron centric, but glia is such an amazing cell, such an important thing that I know, of course, is I keep on telling my students that we need to address both types of cells, but we always look at, the synapse and the synapse, but oh
yeah,
my,
it's a lot of low-hanging fruit.
Yes, and the other thing I want to comment is that you show such a beautiful data, and it's such a beautiful story, and looks so easy, but it's not. It's a lot of work.
Yeah, it's a ton of work, and I should say, that postdoc, he received his training as a graduate student he studied calcium signaling in very early zebrafish development, and so he was really just the perfect person for this project at the right time.
Wonderful. I'm glad that you found him and he worked with you.
Me too.
Lovely to have students and trainees that really love the science, and very, very amazing. So we have a couple questions for you. And we have one of our students, Cielo, she's asking, so considering norepinephrine induces the dynamic calcium signaling astrocytes, can we hypothesize anxiety stimulates astrocyte function?
That is a great question. So does anxiety stimulate astrocyte function? We have not directly tested that, but so I could say, yes, you can assume that. Some folks have done really cool work looking at behavioral outputs and how different behaviors affect astrocytes. In particular, there was a beautiful paper last year from Misha Ahrens, published in Cell, looking in zebrafish, so not at the spinal cord astrocytes, but this other class, they call them radial astrocytes. So we think they're doing what astrocytes do, but in a different part of the brain. And those were responding to like frustration and futile movements. Anxiety in particular hasn't been looked at in the fish, but that would definitely be something that you could test. Like you could make the fish think, there's little behavioral tests you can do, make a spot get bigger and bigger and bigger, and so it thinks a big fish is going to come eat it, and so that would definitely induce anxiety. It would be really cool to look at the astrocytes during that behavioral test, modulate the astrocytes during that behavioral test, and see if that changed the behavioral output in any way. It's a great question.
Great, wonderful. Then there's another question. He says, Edwin says, was there any bioinformatics approach, and where did it help?
So the bioinformatics, we have not, you could look at, so the aqua analysis that Kira Paz Cancer's lab developed, that is a bioinformatics approach to taking sort of the pixels, the intensity of the pixels, and translating that into sort of quantifiable astrocyte behavior. And so that allowed us to go from, which is a super cool movie, I think, but go from a cool movie to actually describing that movie in a mathematical way. The other way that bioinformatics has helped our studies is the single cell RNA-seq studies. So those are actually all done by other labs. There's about 3 or 4 really nice resources out there of zebrafish, particularly zebrafish brain single cell RNA-seq at different time points. And so based on those publicly available data sets, Jack Kuhn was able to do bioinformatics approaches to find clusters of cells that most likely corresponded to the astrocytes that he found. And that let us look at other markers in a more targeted way. And that same bioinformatics approach will be really useful for us as we move on to new CRISPR screens and try to prioritize what genes we should target.
Okay. Jose Enrique.
Yeah, I have two questions. One is technical, and the other one is more like morphological. What is the, is there any interaction of the astrocytes with the ependyma? Because it looks like they were coming off the central canal.
Yeah. So we have not looked carefully at interactions with astrocytes in the ependyma. I can tell you that this Glast also does label some ependymal cells. And so there's a woman in Paris named Claire Wyatt, who's really interested in sort of mechanosensation in the ependyma. And so we have been talking to her about this, and I think we will just send her the fish and let her look at it because that's really her area of expertise. But yeah, good observation. It does look like that.
My other question is technical. It's like, why those so few cells are labeled?
Yes. So in the stable transgenic line, every cell is labeled. But when we do the single cell sparse approach, that's just luck. And so we've taken that DNA construct, just injected it into the fertilized egg, and wherever the clones go is where they go. But we do that so we can see the single cell resolution.
Thank you.
Yeah.
Okay. Now, I had a question about norepinephrine as well. What is the mechanism that norepinephrine is doing for getting calcium in the glia? Is there
I do not there are receptors on the astrocytes, but exactly how that gets transduced into the second messenger calcium, I think is still a little bit murky, even in the mammalian field, and that there's some conflict. People don't know what calcium means or does for the cell, other than maybe like a couple of transcription factors, right? And who knows what that even means. I say that's something that's still an active area of research, even in the mouse, even in the mammalian fields, and I don't know the answer.
I was quite intrigued by that, because then I thought, okay, so there must be alpha or beta receptors, and they must be attached to a intracellular cascade, and then come kinase, and all these things that I have already know in the neuron. Perhaps it may be present in the glia.
Yeah, they do possess the receptors, but exactly the signaling cascades, I don't know.
Okay. Well, any other questions before we continue to the recruitment?
Well, doctor, can I say something?
Yes, Cielo.
But I think it's still on, right? I think it will also be very interesting to see other neurotransmitters that would modulate the astrocyte function, because right now we are observing the norepinephrine with the stress, but if we can observe this with dopamine, for example, we can also observe the function of astrocytes in problems like addiction and such.
1000% agree with you. Yeah. I mean, looking at one piece, right? It's like reading a book with one letter. You have to look at everything. I totally agree with you. That would be really cool experiment and very easy to do in zebrafish, because we can just dump it on the water that the fish live in.
Yeah.
Amazing. Thank you.
Thanks.
Good. So,
okay.
I
will
continue to the other.
Okay, I go back to share the screen, and then I'm going to talk just a little bit, but then I'm going to turn it over to Mateo as soon as I can, because he can tell you a lot more about a trainee's perspective living in Portland. And so, okay, let me close that. Okay, so you can see the slides again.
Yeah, we have
Can you?
there you see. Yes, Portland.
Okay, so Portland, yeah. So we're located in Portland, Oregon, on the west coast of the United States, in a region of the country called the Pacific Northwest, or Cascadia. This is a view into the hills, this old town, downtown Portland. It's a beautiful city. So I was talking in the beginning, I'm from Ohio, which is exactly in the middle of the United States. It is the flattest place you will ever visit. We have a lot of corn. It's not, I mean, it's home and my grandma's there, and so I still love to visit, but this is absolutely the most stunningly beautiful place I've ever lived. So this is Mount Hood every year, which is also a volcano, but don't worry, it's not going to go off anytime soon. There's a lodge up here that we have our retreat every year, and this only takes about an hour and a half to drive to. The city of Portland is really fun and very vibrant, very progressive, lots of great food, lots of great things to eat and to drink. We don't have a baseball team, which is a bummer because I love baseball, but we can go to Seattle for that. We do have a great soccer team and a great basketball team, which is a lot of fun. And the outdoors are just amazing. So there's waterfalls everywhere, hikes. This is my dog at the beach. So the beach is only about an hour and a half away. The water's very cold, but it's very beautiful, just a very stunningly beautiful part of the world to live in. Okay, so this is where we are. Who are we and what are we? So I'm going to tell you about a couple of different training opportunities in neuroscience that we have at OHSU. And I know this is being recorded. Also make the slides available to anyone for the information and the websites and email addresses that are in here. So I'm going to talk first about the neuroscience graduate program. This is our PhD one of two PhD programs in neuroscience. There's also a behavioral neuroscience program, which I'd be happy to connect anyone with, but I'm going to focus on the neuroscience graduate program. That's the program for which I am the director. And our goal, so this program was founded in 1992. I think I am the fourth or the fifth director in that time. I'm the fifth. And so we aim to train pre-doctoral students in modern neuroscience concepts and techniques. And some of our program strengths, I think, are the diversity of our research community, the training that our students get, and the career prospects after graduation. And so, like almost all, I think all graduate programs that I know of in the States anyway, we cover all tuition, fees, and insurance. And by the time, if you're a senior now and you would come next year, the stipend would be about $35,000 a year. And Mateo can tell you about how easy it is or isn't to live on that in Portland. Our areas of research strength are molecular and cellular neuroscience. So as I said, there's a behavioral neuroscience program as well. There are many folks that overlap in both, but we are focused on molecular and cellular, neuronal signaling, gene regulation. So kind of a lot of the stuff I just told you about. We have a lot of strengths in biophysics of channels and transporters, sensory systems, developmental neuroscience. And there's a lot of us that work on glial biology and neuron-glial interactions, which is one of the reasons that I moved my lab from WashU to OHSU. We have a lot of faculty, so more than 140 faculty members that you could rotate with, the potential mentors. And there are many different institutes, and this is on a timer, sorry about that, institutes and departments that have PIs with whom you could rotate or join. So the Vollum Institute, you can think of the Vollum Institute as a neuroscience department, basically. Oh my gosh, I'm sorry. As a neuroscience department. So this is the Home Institute. This is where my lab is. And this is where the funding comes from for the neuroscience graduate program. We also have a primate research center, a hearing research center, a center that's really focused on neurodegeneration, occupational health sciences, as well as many basic and clinical departments like neurology or the Department of Behavioral Neuroscience. So it's a great really collaborative neuroscience community that spans lots of different departments, institutes, and centers. So we have currently 54 students. We have approximately 10 new students join each year, and the average time to degree is 5.2 years. One thing that's unique about the program is that the core courses all happen in the fall, right after you arrive, and then the rotations happen in the winter and spring, so that you don't have competing. You can focus on your classes full time, and then you can focus on your rotations full time. We really, really care a lot about mentor-mentee match, making sure that you're joining the lab not just because you're interested in the science, but also that this mentor is the right mentor for you, and that this lab environment is the right environment for you. And so we think that by allowing full-time research rotations, where you don't have your focus pooled by classes, you can really get a better sense of if this is the right mentor for you for your PhD training, because you're going to be in that person's lab for, over half a decade. And so you want to make sure that you guys, that together you're the right fit. One thing I'm really proud of, this happened before I arrived, but one of the things that I've always loved about this program, and Matteo will tell you more about this, is that at OHSU, our students were instrumental in founding the Alliance for Visible Diversity in Science, which is now a campus-wide group, but it really started as a grassroots group in the NGP. And many of our students go on to careers in academia, but also industry, government policy, science writing, and we keep a running list of all of our alumni here on our website. And so along with their contact information, so that if there's a particular field you might be interested in, you can connect with that particular alumna or alumnus and start to network in that field. Right, so that's our grad program. I am so lucky that we recruited a phenomenal coordinator, Ms. Jessica Parks. She has a master's in education, and she is just, Matteo can tell you, a wonderful resource for the students, really fantastic. She takes care of really your day-to-day needs and your day-to-day lives, and is just a great person to work with. Here's our website of the volume, the program website, as well as all of our faculty. So that's the graduate program. That's to get your PhD. OHSU also has a great MD PhD program. So I don't run this program, but I can provide you with a little bit of information because it's really fantastic. And if you came to the MD PhD program, you can also join the NGP for your graduate training. So the students come 18 months in the MD training, 3 to 5 years PhD training in any program you would want, including the NGP, and then your final 2 years of the MD training. And again, everything is covered: medical school tuition, fees, insurance, graduate school tuition, fees, insurance, and the stipend is the same, about $35K a year. So if you want more information about the MD PhD program, you can contact Alexis Young. She's the program coordinator for the MD PhD program. She's also phenomenal, and she also hosts monthly information sessions, so you can email her about that. I'm really sorry about the timer on these slides. I did not know that was there. Okay, so those are our post-graduation opportunities for MD, PhD, or PhD. But in case you want to know more about us before that time in your career, we also offer a few opportunities before graduate school. So one is our Summer Undergraduate Research Program. This is directly through the volume. NGP. There are other research opportunities available for summer undergraduates at OHSU, but this is ours. It is an 8-week program that runs from mid-June to mid-August. We cover your stipend, a stipend that covers housing and living expenses, and in addition to working in a lab full-time, and we also try really hard to match you with a mentor who's right for you both scientifically and personally. So you're here for the summer. You have a full-time research project in the lab, but we also have classes and didactic sort of mentoring on applying to graduate school, helping you with your applications, reading your essays, and the like. applications for this close in early February. And we do, I mean, I know COVID's happening, but we do expect this program to go on next summer, even if, the pandemic's not over, we would transition at least part of this to be virtual. And so here is the website for our summer undergraduate program. A couple of years ago, Jocelyn Santiago Perez was one of our summer undergrads. Here she is on a hike. She worked in the Westbrook lab and I believe had a great time. She's taking a gap year right now and applying to grad school soon. Great. And then finally, we also have a year-long neuroscience post-bac initiative. And this is a newer initiative. It's in its third year now. This was our class last year. Denisha King is now a first-year graduate student at the University of Rochester. Raquel Moreles is now a first-year graduate student at the University of Virginia. And very happily, Jessica Santana stayed at OHSU, and she's now a first-year NGP student. And so this was launched as a collaborative effort between a couple of departments, ours, behavioral neuroscience, as well as the senior vice president for research. And this is specifically targeted to provide sustained, paid, year-long research experience and skills for individuals from underrepresented backgrounds. And so you work in a lab full-time, you can take classes, you can take any of the graduate courses you might like. And there's also mentored research experience, mentor-mentee training, and really tailored individual support around academic and professional development. Here is the website, and these applications close on March 1st. And before I turn over to Mateo, I'll tell you also about a brand new initiative that was just launched on November 8th, so very, very recently, at the Volume. And so this is our new Center for Racial Equity and Inclusion. And this is run by Dr. Antoinette Foster, who's a recent graduate of the Neuroscience Graduate Program, as well as Dr. Letitia Wyatt, who's a professor in the Department of Neurology. And so this is a very new center, but what we hope is that working together with Dr. Foster and Dr. Wyatt, the Volume community and the NGP community, we really want to work to dismantle individual and systemic racism that affects our faculty, our staff, our students through really data-driven procedures and policies, and really focused and sort of initiated at the level of community building and education. So really looking forward to continue to work with both Dr. Foster and Dr. Wyatt and evolve our policies along these lines. And so with that, I am very happy to turn it over to Mateo, a fifth-year student in the program, who will tell you about life in Portland, as well as our student group, AVDS.
Hello, guys. Can you guys hear me? Yeah, it seems it's working.
And you can tell me to advance the slides.
Oh, yeah. Perfect. So, hello, I'm Mateo. I come from Colombia, but I'm here right now in Portland. I'm going to tell you everything about what it is to be some like clueless Latino dude coming into America and living the dream of neuroscience. Woo! So before that, let me talk about the AVDS, which is the Alliance for Visible Diversity in Science. We started as a student-driven group, started by Antoinette Foster, now a doctor. And we are essentially working towards racial equity and inclusion. Can you pass the next slide? So, there is two ways in which you can tackle a problem, and this applies also for neuroscience. You can go like bottom up, looking at cells and how they interact in circuits, and further on and greater, or you can go top bottom. So you can go into psychology and see how this interacts with certain parts of your neural activity with fMRI and so on. So the same happens working with equity and inclusion. You have to tackle this problem from both sides simultaneously. So top to bottom is to change policies, to change the environment and the structures that allow or don't allow for like racism and white supremacy. And the other side is to change the zeitgeist, the attitude of people, and that's done through community building. That's done by just hanging out, speaking your mind, learning from others, having empathy, and just being friendly. So that's the two parts. That's the two-pronged approach of AVDS, so community building, policy change, and I can attest that both of them are going on. So next slide. Community building, which is what I do. I am the chair for events or party person, however you want to call it. We have a bunch of cool activities in which we used to like come together before the quarantine and the ends of times, but nowadays we just do it virtually. Nonetheless, we have book clubs, we have happy hours, we have like recruitments of for the different graduate programs that exist in OHSU. We have invited speakers. We share important resources and information about racial equity and inclusion. And all of these, it's always just trespassed by the notion of learning, like of having a little bit of empathy, of putting yourself in the shoes of others, of like deconstructing your own assumptions, and then just being vulnerable and having time in doing so. And I can say that personally, coming from Colombia, where there's not a lot of racial diversity, at least in Bogota, and where the history of slavery and racism was entirely different as the United States, I had a bunch of preconceptions and assumptions that were here happily deconstructed, and I think now I'm a less bad person. So that's one side. That's like the fun side. But there's also like the policy side, which is also fun, but for other people. So next slide. AVD is a really big group. We have a bunch of other talented people working on interacting with the administrations within OHSU and figuring out how to change these structures, how to change policy so they are more, yeah, they are better into like bringing and retaining and making people from racial minority, like underrepresented minorities feel home. So a bunch of proposals were done a while ago in 2017 and 18, which consisted in like creating a new dean for diversity that's just overlooking all of these efforts from OHSU and having a direct line of communication with the presidency and so on. Also changes in how like funding is being provided for OHSU for like speakers of coming from underrepresented minorities, like because science should be across all races, and how to like keep track of the data of students that come here and like they for some reason don't stay, or why do they stay, or how they feel and how things can improve, because we are expecting to have decisions driven to make decisions driven by data, not by speculation. So overall, these many of these proposals were, well, these are some of the proposals that we made and were adopted, but it's racial inclusion. It's an ongoing project and it's an ongoing process. So more recently, and what Kelly was mentioning, and after all the turmoil that came with the killing of like the police brutality during the summer, AVDS took a stronger stance towards dealing with its own structure that might perpetuate racial inequity. So with that in mind, now Center for Inclusion and Diversity has been created, like these REIs that Kelly mentioned earlier. And the purpose of that is to just expand these overlook and support network towards all of OHSU and really invest on that so people from underrepresented minorities can feel at home. So next slide. So we are a relatively big group, but if we are working towards deconstructing these structures of dominion, we also have to, it would be hypocritical if we had like a pyramidal structure. So we are kind of a donut structure, which is far more delicious. So we're essentially a board that has the chairs of different parts of the organization. We have communication, events, education, policy, faculty, and operations, and you can divide them both in policy or community building, and we just like keep track of events in the campus, and we interact with students and with administration, and we plan these events, and we inform policy, and all of this is done by a huge and wonderful team of volunteers that are also supported by receptionist, treasurers, and secretarian. So long story short, we are here for you guys, and if you come here and you want to join, please do. So next slide. We have been growing through the years. We started as a student group, as Kelly mentioned, initially by Dr. Antoinette Foster when she was a graduate student, but now the group has grown a lot, and we are far more heterogeneous in terms of the people that composes us. So we have more points of view and more ways of interacting with the different parts of the university, and we have been growing throughout the years. And I think that's that on this. So yeah, you guys can check us out. And yeah, that's pretty much it. I can now speak on behalf of all graduate students, because I am a representative sample of all graduate students in OHSU. Not really, but I can give you a sense of how life is in Portland as a graduate student. So let me start by saying that when I first knew that I was gonna come to grad school, that was the idea. I said, okay, I literally Googled best places in the United States to live. I'm not kidding you, because science is horrible, and of course, this is more to that. Like, it's beautiful as an ideological thing. It's like the search of truth and all these wonderful interactions, the neuroscience, how the mind works, and so on. But then when you come to reality and you come to the lab and your experiments don't work, it's hella frustrating. So it is important to have like a nice space outside of science in which you can live, because you are a scientist is not a person in the lab. A scientist is an integral human being, and you need to like be in an inclusive and wonderful space where you can nurture yourself and like put all those good intentions into your science. So that for me is Portland. I came here entirely clueless. This was the first time that I was living for so long outside of Colombia. I had a broken English. Now it's not that broken. It still has a little bit of an accent, but people think that I'm from, I don't know, Italy because of the name or some stuff like that. And I found a bunch of wonderful people. I found a bunch of wonderful friends within my classmates from my year and all the other years. I found housemates within them. So going back to what Kelly was saying, how is money-wise living in Portland? Well, if you have housemates that are awesome like mine, and it's not bad at all, and you can also live alone, and it's not an issue. So yeah, I came here and I found science, but more important, I found an environment that it's both fun and pretty liberal and relaxing and supportive, and a bunch of good friends and interactions. And the nature, you guys saw the pictures that Kelly was showing you. I personally I'm from Bogota, which is high up in the mountains. It's like 2,600 meters over the level of sea. So I'm a montañero, unlike you guys. But still, you can just like drive 1 hour and a half in the other direction, and you're in the ocean. So it's pretty neat. The city itself, you have a lot of things to do around. There is many neighborhoods. You can live in big buildings, or you can live in like a suburban house. There is many activities to do. If you like sports like I do, you can just come climbing or like basketball, or there's like intramural league of soccer and so on. There's like all sort of things that you can do. Yeah, that's pretty much it. I cannot speak about my science because I don't have my presentation with me, but I can tell you that it's working insofar as my life in this place, it's working. And yeah, this smile is real. So if you guys have any questions, just go ahead, and I'm here to answer them. I can both speak in English and Spanish, or Spanglish to make a compromise. So go for it.
Preguntas para Mateo. you can unmute yourselves, or to Kelly in English or Spanish. You have that opportunity. ¿Alguna pregunta?
Well, I was going to ask you about, because you mentioned you had undergraduate research opportunities, but aside that, do you have other undergraduate opportunities in the neuroscience department?
I'll take it. Sorry, Mateo, to cut you. The Volume Summer Undergraduate Program is specifically for neuroscience. There is another summer undergraduate program called Build Exido, and that is not necessarily neuroscience. That would encompass any of the programs at OHSU, which includes cell biology, chemical engineering, bioinformatics. There's other graduate programs besides our own, and so Build Exido would have, if you were interested in other areas as well. But for the neuroscience, ours is specifically focused on neuroscience.
Okay, especially like psychology. How would you place a psychology student in your track?
So that may be more appropriate historically for the behavioral neuroscience program, I would think, which is run by a woman named Marina Guzzeri. It's Italian, and I always say it wrong. But she's fantastic. That is run out of the behavioral neuroscience department, and whereas our historical focus has been molecular cellular, their historical focus has been more psychology, but I think now both programs are quite interested in systems. So there's overlap, but also distinctions. But I'm happy to introduce anyone to Marina or students from that program as well.
And they do have a summer program, the behavioral neuroscience, or
not their own, but if you were interested in the Volume Summer Undergraduate Program, you could certainly be in a behavioral neuroscience lab, and many faculty are jointly appointed in both programs.
Wonderful. Where do I find the information for that program?
I'll put it in the chat. I have to open up the PowerPoint to find it, or it was on the PowerPoint slides, which we could make available if that would be easier.
Yeah, well, I have your presentation with me, so I'll share it with everyone. Yeah. It will be.
And I will.
I'm sorry.
Oh no, sorry. Go ahead.
It will be available at the Neuro-ID website as well. So if you want to look at the slides, you can do that. So no problem. And there's Ian has shared the link.
Thank you. Thank you.
As well.
And,
great, thanks.
And also, I put an email address in there. That'll go straight to Jessica Parks, and she is a contact. She's a good starting contact person for anything. So that's just ngp@ohsu.edu, and she could direct you to the correct faculty member.
Okay, great. And then there's Edwin asked, what's the acceptance rate of the programs, both the summer and the PhD?
Gosh. So last year, I think the neuroscience graduate program received more than 200, but less than 300 applicants. I do not know the exact number. We interview between 30 and 40 every year, and accept about 25. And we hope to have a class of between 9 and 12, 15. It's kind of fluid. One nice thing about being at an institute is we have our own endowment. And so when I took over the position, I actually asked my boss, what's my budget? And he told me, I will tell you when you're spending too much. So, so I took that as a personal challenge. And I haven't been told.
Oh my God.
I haven't been told yet. So I mean, from maybe 250 to 45 to 25. For the summer program, I actually do not know the acceptance rates because I have not been. We host students in our lab, but I am not part of the committee who reviews. That's separate from my jurisdiction. I don't know.
We can, if we have students that want to apply to the SROP, we certainly will let you know so you can,
Great.
usher them to make a good, application. So it will be a competitive application, so they could, be able to get there somehow. So
Yeah, great.
and Mateo, there's a question for you.
Yeah.
I think.
Yeah, I'm reading it. How's the Latin experience in Portland?
¿Cuál es
Well,
la experiencia de ser latino en Portland?
English or Spanish? Spanglish. Okay.
En
So Kelly can
español,
laugh along.
Kelly won't mind if you speak Spanish.
Je parle français, mais that's it. Sorry. Yeah, go. You do whatever you want.
Okay, so I would say that it's pretty positive overall. Within OHSU, when I first arrived, I was kind of one of the few people that spoke Spanish, but it has like got better with the years. That being said, outside, that's talking about only the NGP. Outside of the NGP, there's plenty of Latin people around. I have friends that there's big companies around. There's like Intel, there is Nike, and all of these companies have also like a huge, yeah, they attract a lot of brains from elsewhere. And I have a handful of friends from Mexico, Chile, Colombians. Oh my God, or a plague. And overall, I think if you look, I haven't not looked for it actively. I don't have that drive in me, but I've seen it just secondhand. Like whenever these friends would just take me to, I don't know, Aztec Willis, which is this like Mexican place where there's like salsa nights every Friday. We'll just go there and see a bunch of people just like murdering the dance floor with all their moves. And I'm from Bogota, man. We don't dance. We're bad. We're bad Latinos. But to make the long story short, yeah, like there is a lot of Latin people around here. So if you look for it, you will find it for sure.
Okay. Okay. Any other questions?
I had a question, but this is more like research related. I'm sorry I didn't do it earlier. But would you also recommend the zebrafish for epigenetic research?
Yes. See, so I am a model organism proselytizer. I firmly believe that you should, well, I like to study the simplest animal possible for the question that I want to ask. And so there are a lot of really cool epigenetic labs that have zebrafish as a model system, particularly with like regeneration, heart regeneration, limb regeneration. There's a really cool lab at Duke who works on that. One at WashU, she works on spinal cord. So I think, fish, not great for lung development, but if you want to understand fundamental mechanisms of most biological processes, I think starting simple and showing that conservation. So if I find a gene that works in fly and it works in fish, I'm pretty sure it's going to work in humans, right? And so yeah, that's why I like working in simple things. But yeah, there are a lot of really wonderful labs using zebrafish to study epigenetic regulation of lots of different biological processes.
Great, this has been a wonderful seminar. Thank you.
Yes, it has. Any other questions? Because we're reaching our time to say goodbye. So I just wanted to see if anybody else wants to. Doesn't look like it. So Kelly, Mateo, it's been a pleasure, a real pleasure and a treat. Everybody said it has been wonderful, and I really appreciate your time and sharing with us your wonderful science, and also your enthusiasm of creating a diverse community that makes sense, and I think that it totally in a world where we have globalized, ourselves such a way. Transforming that into our own scenario is such an important thing, and reaching out to creating a very welcoming and equal environment, I think is such an important factor. And the fact that you have an institution that supports you in such a way is wonderful. So I have to tell my students that this is an excellent opportunity, and you all should consider a Oregon Health Sciences University for summer or grad school, whatever. It's wonderful. Thank you. And so, as I said, next week we don't have a seminar. We start again on the 2nd of December, which is a Wednesday. Well, we'll have another presenter, ¿verdad? And this time is our former student from our institution that now he's at MIT doing his postdoc, and he's gonna be talking about reopening of juvenile-like plasticity using pharmacological and digital therapeutics in the adult mice. We'll listen to Jesus then. So thank you so much, and have a wonderful week and happy Thanksgiving to all of you. Thank you.
Just before I forget,
Mateo, sí.
it never gets too cold. That's so important. That's so important. It's rainy for sure, but it's not like freezing cold. And as a tropical person, I highly appreciate that.
Yes.
And no mosquitoes.
No mosquitoes. That's applause.
No.
Okay, well, thank you so much, everyone, and I'll see you in 2 weeks. Okay.
Thank you. Thanks for the welcome, and thanks for all of the really fantastic questions. Thanks, everybody.
Thank you, Kelly. Adiós,